All right, so let's start by setting the scene. So let me introduce to you Sir David Bruce. This man was a Scottish microbiologist who played a key role in understanding neglected tropical disease today. He was part of Britain's Royal Army Medical Corps and served as the leader of the 2nd and 3rd British Royal Society's Sleeping Sickness Commissions in the early 1900s. And these commissions were led to investigate a phenomenon of strange disease happening in Africa. So him and a team of researchers decided to leave London and went on a journey all the way towards South Africa. And you can see his team right here, all 5 of them. And most notably, there in the back, right behind him, is his wife, his greatest scientific collaborator, Mary Elizabeth. And they co-authored 174 different publications altogether. And when they arrived to Sub-Saharan Africa, they were presented with this disease. People were sick. They were stuck in this state of somnolence. They were sleeping, they were lethargic, they weren't moving, and they were just, it was undescribable. It was like a world that you haven't seen before. And this is what we call human African trypanosomiasis, also known as African sleeping sickness. Every single one of these individuals were displaying this unique Winterbottom sign, in which the lymph node around the neck indicates that the individuals were bitten by an infected tsetse fly, and thus there was a causative agent to this disease. And not only were the humans affected, the cattle were affected as well, presenting a disease known as nagana. Here, the cattle became highly lethargic, they were weak, they were losing a lot of muscle mass. And unfortunately, for both these humans and animals, without any medications, the fate of their survival would be nothing. They would all die. So, Sir Bruce and his team discovered the causative agent for this disease, and this is a unicellular eukaryotic protozoan parasite known as Trypanosoma brucei, as seen right here. This is considered a salivarian trypanosome, meaning it is transmitted from the bite of an infected tsetse fly, as seen right there. There are 3 different subspecies of the parasite that can be transmitted. The first, T. brucei, which causes a nagana disease. Then there's also the 2 forms of HAT which can be caused as well. The first is transmitted by T. brucei gambiense, causing a more chronic, slower acting version of the disease. And secondly is T. brucei rhodesiense, which causes a more fast acting, deadlier, acute version of the disease. Now, across Sub-Saharan Africa, where you find this disease is, for the chronic stage at least, is primarily within Central and Western Africa, while in the eastern parts of Africa you find in blue the more acute variation of the disease. And as our society, as our worlds are becoming more connected, we see increased rates of transmission and outbreaks in places and other countries that you wouldn't expect. And with climate change happening, we see the tsetse fly being able to harbor and survive itself in other parts of the world as the weather gets warmer. So, over this duration, since the initial discovery to today, there have been many, many different methods of controlling and mitigating the spread of this disease. The first is through tsetse fly management, where big nets are used to capture the populations and control them. Secondly, there's this technique known as vector sterilization, where scientists in labs will grow massive amounts of male tsetse flies, and they'll all be sterile, which they then release into the environment, and when they mate with the females, the offspring will be non-existent because they're sterile. In addition to that, we can also have a more human-centric approach. We can educate the people at the highest risk communities to ensure that they take the necessary precautions to avoid acquiring the disease. And of course, we can, if individuals are to be afflicted with the disease as well, we can perform population screening to ensure that we catch the disease early and we can apply effective medications. And as such, there have been numerous drug programs established over this course of time through nonprofit organizations such as DNDi, which ensures and helps funding of research of numerous different medications, which are continuously being released every few decades or so. And with this, over the course of time, the disease has decreased. This graph shows over the past 2 decades, thousands of individuals are not being affected, are no longer being afflicted with the disease anymore. But unfortunately, with changes in governments and axing of international collaborative programs to assist developing countries, they're being gutted. And as such, we lose the loss of health data and monitoring. We have less purchasing power for those essential medications to ensure that people don't die. And the question we ask is, are we doing decades of progress? Well, do we give up? Is this it? Do we just take the L and continue forward? I say no, and myself and many others. So let's talk a bit about the research that I do. Let's talk trypanosome biology. So the way that this parasite works, let's get a little complex here, is it does something called antigenic variation, in which the parasite is coded with a single antigen. We will call it the VSG. In a normal sense, the antibody of a host will bind and recognize an antigen, in this case, the VSG. And the antibody will then clear the infection and kill the parasite. However, this parasite has a trick up its sleeve in which it presents that single VSG code. It alerts the immune system. It says, hey, come here and attack me. But then in that time before the immune system is able to develop an effective response, it changes the code and hides in plain sight. Because of this, because the parasite is consistently changing its antigen, there is no effective way to make any sort of vaccine in this current day. So what my research is I want to understand the biology. I want to understand the proteins and genes that cause VSG switching to occur. So we go and we ask the question, what are some proteins that might be involved here? How can we modify the parasite so we can better study it? We can then take a whole bunch of different genes and modify them with a whole slew of techniques to create different assemblies. And we can integrate these assemblies and put them into our parasite, essentially changing the way that it works normally. We can then take our modified parasite and perform a bunch of tests, different assays, and see what can occur. And based on the results of these assays, we can then learn and we can write our findings, and over the course of time, we'll make newer and greater scientific discoveries. So how does something like building with biology look? So let's look at a very simplified example here. So for example, we can have a construct at the bottom here, which has a protein tag, which we can insert at the end of a gene of interest. So when we have this protein being expressed, we can then integrate it into the genome of the parasite so it can continuously be expressed, and we can study that specific gene and thus protein to be studied. For example, we can do something called immunofluorescence assays, where we can light up the protein of interest. So in here, I'm not too sure how well you guys can see it, but there's these green dots, and these green dots indicate this protein that I have tagged, and we can see it is localizing in this blue section, which is the nucleus of the parasite. In addition to looking at it under the microscope, we can actually build a network. So through an experiment called immunoprecipitation, we can take that protein and we can concentrate high amounts of it. The benefit to this is not only are we getting our protein of interest, but all other closely interacting proteins. We can build here a network. We can take that protein and study it and map it and better understand what is going on and how these proteins are interconnected together. So, for example, a figure that I have made here, we can demonstrate the protein-protein interactions through what we like to call an interaction network, where we have a central protein right here, and we can see a whole bunch of different proteins here, all coming together and having some sort of closely associated interactions. In addition to that, all of these proteins have functions. So we can perform something called a gene ontology analysis, a GO analysis, and we can see, for example, what are the similar traits within all these proteins. So for example, here we have over there in that big red circle, we can see RNA processing. This is a very, very key function that we can find with all of these proteins here that we can better understand and target, so we can have a greater understanding of how VSG switching is related in contrast to RNA processing. And lastly, one other analysis we can do is do protein comparisons between other different species. So for example here, we have the AP1 protein of our parasite, our Trypanosoma. And we can compare it against other species in humans, in plants, and in yeast. So we can see which parts are more conserved and which are less conserved. And this will give us some insight as to which sections we could potentially target. What kind of therapeutics can we design to target non-conserved regions so we don't have these drugs accidentally attacking us as humans and only killing the parasite. So that's it for me for now. I would just like to give you some 3 key takeaways. And the first is that global efforts to mitigate HAT have been super effective for now. And unless we continue to take the initiative, form these developments, this might not be the case in the future. Additionally, for science, protein networks highlight these interconnected proteins of interest, which we can better study and have a overall better understanding of how these complicated immune-avoiding systems work in these parasites. And also, lastly, we can perform comparative alignments between our parasite and proteins of other species, so we can find other potential therapeutic targets in the future. So yeah, thank you.